I think it's, in my career, this is the best thing that happened to me in my career, being able to be in Larry's lab. And as Scott said, just a community. There's just a community of geneticists. Yeah, I feel like I'm part of a club. We don't have a secret ring or handshake or anything, but yeah, oh, great. So, yeah, the title, what I'm going to talk today is great. It follows Steve's talk. I'm going to show you that chromosomes can do just fine without a centromere. So that's what I'll talk about. So why did I have this title? And it just relates to Larry. This was a time in the 80s there when molecular biology was just sweeping through the labs in the country, and Larry blithely ignored that. And I just really like that somebody, it was coming up and talking in the lab. We were talking, why don't we do molecular biology? And this was Larry's comment, why walk when you can take the meiotic drive? And that was a time when you're walking through chromosomes. The other thing that I really like, Kent said this. This came up. Larry, you view Drosophila simply as a vessel for transmission of the chromosomes. And do you know what Larry answered? This was perfect Larry. That's not true. I view Drosophila as a vessel. And so that was wonderful. So, and let me just go back to how I ended up in Larry's lab. I feel really fortunate how I ended up in Larry's lab. If I go fully back, it's because of Jacques Cousteau. So I grew up in the era with Jacques Cousteau on every Sunday night, and all of us I went to UC San Diego because they had a marine biology lab, and everybody else at UC San Diego wanted to be a marine biologist. And so it happened, I don't even know how it happened. So I worked in Scripps doing fish fins or something like that. But at the same time, I can't even remember how but at the same time I had an opportunity to work in Dan Lindsley's lab. And I had just taken a undergrad genetics course with Bruce Baker and Natalie Carpenter. And Pat Romans was my TA, and she's in the audience someplace. Actually, so there was this amazing paper. So Dan, said you can come in the lab. I work with Jim Kennison. He said, read this paper. And as an undergraduate, this was, and it was really great, though. Jim Kennison walked me through it, and eventually, after a year and a half, I think I got it, maybe. But this paper really, as people have said, is truly foundational and really decades ahead of its time. It's the first functional genomics project, and still it stands as that. And at the time, I didn't realize, there's Jim Kennison. I was an undergraduate, so didn't realize all these people, but Jim Kennison, Natalie Carpenter, Bruce Baker, Jim Crow had done a sabbatical there. I took his class. So it was really, I didn't know this, but it was quite an amazing group. But all during that time, Larry's name would come up. And Dan Lindsley, to me was already this mythical figure, but there was this other mythical figure up north, and it was Larry, and Larry's name would always come up. So when I decided I have to go make the pilgrimage and go up there. So I went to Seattle to the genetics department and to work with Larry. And so, and I think really we've talked about this Larry's chromosome course, and it was really quite amazing because it doesn't sound like chromosome mechanics be interesting, but Larry had a way of making this the most interesting thing. And I don't have this quite right, but here's what I remember just in the beginning, his introduction to the course. He's like, we'll start at the centrosome and wend our way to the telomere. Along the way, we'll encounter many wonders of the chromosome. And of course, as geneticists, we'll proceed from the formal to the physical. And, and so in that too, he made a point that we're geneticists. So all of a sudden, we're, second-year student, I'm a geneticist, and he would sort of indoctrinate you with that. And that was really wonderful. The other thing I remember, so as I said, in Larry's office, we would go through the crosses and everything. And one thing Larry would always do at the end of a cross, I don't know if you guys remember this, he would put up, he would always make a $ sign, that was the final, the result you want to get. And that always interests me because Larry, out of all people I've ever met, didn't have any interest in money. So I always found it ironic that he would put the $sign there. And I think just one story about that, I think Larry, is the only one in the history of the NIH to turn back NIH funds. So anyway, he didn't spend all the money, and he turned it back, and he took great glee in the fact, he viewed it as a genetic experiment. He took great glee in the fact that they didn't know what to do. And so he'd come into the lab and say, yes, the whole NIH process is for dispersing money, and they don't have the reverse process, and I've proven that by trying to turn it back. So he was really took glee in that. Yeah. So this, I went to my notes, Larry's notes from his course, and here's Larry's rules of crossing over. So he has these 10 rules of crossing over, and I guess I have it here. So here's my handwriting here. 10 propositions that enable one to convert crossing over data into exchange data. And so this was Larry. This was, just the beauty of genetic analysis that had been talked about a lot here, where you would take this formal analysis and figure out the underlying biology. And so you learn a lot about what the chromosomes are actually doing just by counting the flies. And so that was Larry's thing. And also I do remember the other thing, if you had a discussion with Larry and he didn't really like how you responded, it wasn't point well taken, it was point taken. And yeah, so here's the Sandler lab, and this is, most of our education. So here's Sergio, this is me, Larry, Kent would be over here. Sorry, Kent. But I do remember this a lot because Larry, whether you were working or not, he would just come in and sit down and pontificate. And just on the history of genetics or whatever, you'd sort of have your flies there and have to quickly put them away and do it. But he would come in and talk about anything. And also I used to, I'd be reading and bringing a book. And what always amazed me is whatever book I had on the edge of my bench there, Larry knew all about it. And I remember this really clearly because I had a book. It was about some folks who just survived in the jungle for months. And so Larry comes over and he goes, oh, that reminds me of Tarzan. So Larry ends up knowing the whole history of Tarzan and why it developed and why it's a myth that stays around even now. And so he would, for anything, so he had an opinion, knew the history of it, he had would have theories of it, and then tell you why of these 3 theories he thinks number 2 is the most likely one. And the other thing, actually, yes, so I was a TA. We were all TAs for Larry. And I don't know if you remember this. This amazes me to this day. So I teach a lot of undergrad genetics. And Larry, when we're walking back from the course, we're walking back, we come by the trash can, and he throws his notes into the trash can. And I'm like, Larry, what are you doing? He goes, well, if I don't do that, I'll be tempted to reuse them next year, and my lecture will not be fresh. And that is still, I haven't yet risen to that level yet. But that's still to this day, All right, and let's see, what else I want to say about, oh, yeah. So this is the lab, and I put this under a dissecting scope. 607 days till the meiotic meeting in Hawaii. So the other thing I just wanted to bring up is, oh yeah, so the other thing we talked about, one time Larry pontificated on genetic load and genetic drift. So Sergio and I made a model for that. So there's Sergio reading a windsurfing book, a skateboard, and there I am working. So, and also the Italians were good at not, I would give them a hard time at not working that much. The Americans were workers. So we had genetic load and genetic drift. But the other thing is Larry in language. This has come up a number of times. Larry was very good, obviously, at speaking and languages. And so he would instill that in us in a number of ways. But if you look right up here, you can't read that, but I got it here. Word of the week. And then I got this example. Bill is Miss, so I have to find what that was. Missologist. Does anybody know what missologist is? Yeah, well, here it is. So that was our word of the week. Missology is defined as hatred. And of course, duh, I can't put that up there. Yeah, of course, yeah. So the other thing I want to tell you about, the other thing about language, Larry would correct Sergio on his Italian grammar. And I'm like, Sergio, is he right? And he's like, yeah. He goes, Larry was, I don't know, Iris, if this is true, but Sergio was saying he learned the grammar first before he learned the vocabulary. I don't know if that's true or not, but he was, he goes, yeah, he's better at the grammar than, most Italians were. Yeah, so let's see. Oh, yeah, so language. So the other thing is, when I gave my first talk, I still remember it was on hybrid frogs, and it was chromosome missegregation in the hybrids. And I gave a lot of jokes in it, and felt good about it. The next day, Larry goes, Bill, can you come into my office? I'm like, yeah, I'll come in. And anyway, he goes, okay, do you want to be a comic or do you want to be a scientist? And I'm saying, and I said, oh yeah, I want to be a scientist or a geneticist. So then, and this came up with, I didn't realize this, that Larry had talked to Ginger Jakian, but Larry said, we're going to, this is not acceptable, the way you talk. We're going to have to do something about this. So he gave me these choices, to be in a play or to take speech classes, right? And so I chose speech classes. So I had to go to the University of, I enrolled in a speech class. The trouble was like, as a second-year graduate student, it was a freshman speech class. I was basically this graduate student with all these freshmen. And so I lasted about 2 weeks, and I came to Larry, and I'm like, Larry, please, I cannot do this, right? I just can't do this. So he said, well, so we had a compromise. I ended up for about a year, I would go into this little booth and read books, and this was for the blind, so I would read all these books. So it was before books on tape. And so anyway, that's what I would do. I read a lot of books for the blind. And I think, I still have issues with my language, but I've improved quite a bit. The other thing about the lab is, and maybe Iris knows about this poster still haunts me to this day. And it's like, and I don't know the origin, but it was there, and it's from the Mideast, it's sort of magical, but there we go. And the thing about the lab, too, was the ether. There was lots of ether, right? And I do remember this. So my first project in the lab, right when I came in, Averill was there. There was nobody else in the lab. I came in, and Larry was doing the Muller 5 X-lethal test to see if Seattle water caused mutations, was carcinogenic. And so I did that with Averill. But even as a second year, and I wasn't known for being particularly cautious, but Averill, with her cigarette in the ether, I'm thinking, this really is not a good thing, right? And somehow we all survived, yes, yeah. So let me get, yes, so finally the final thing is, so this was, I think, important to this. Larry's advice, and it was, when I was thinking about doing a postdoc, I can make you a living pushing flies, but you can't. And I think you see that today. Everybody from Larry's lab has gone off and done other, taken other systems or other approaches and combined them together, and it's actually quite powerful, as you saw today. And at this time, it was really, Bruce Alberts had come through, and I was giving a group meeting, and Bruce, and actually I really have to mention Gerald Schubert. So I really feel like I had two advisors, Larry and Gerald was amazing. So I spanned the two labs probably about equal time, and that was great. So I had two great advisors. And Gerald was good friends with Bruce and invited Bruce to one of our Drosophila meetings, and I was giving the talk, and it was on something with meiotic drive and spermiogenesis. And at the end, Bruce asked a question. He's like, well, what does the actin look like in the spermatids? And I'm like, what's actin? You know? And Bruce was just appalled, and he turns to Larry, and he's like, what do you teach these graduate students here? And Larry is like, there's no reason. That's, we're dealing with the chromosomes. We don't need to know about that. But to Larry, then, like about 4 months later, Larry goes, I think it's just like with Barry, I think you should do a postdoc with Bruce Alberts. And so he did appreciate that this, maybe I should know about actin, and went there. So let me tell you what I want to talk about today. And this is actually a note from my lecture notes from Larry's amazing course. And I don't know who it was, but I have then asked about the fate of a chromosome when you break it. And this is, as Kent nicely described, if you break a chromosome, you can get fusion of the broken ends, you get a dicentric, and you get bridge breakage fusion. And so I think I have that here. Yeah. You break it, you get fusion, and then you get this famous McClintock bridge breakage fusion cycle. Kent was telling me about all his amazing dicentrics and everything. And this was maybe about 5 years ago. I'm saying, well, has anybody ever filmed the bridge breakage fusion cycle? Let's get it live, right? And so Kent said no. And I said, well, can you send me some of your amazing stocks? And I'm going to try and film the bridge breakage fusion cycle. So that's what I'll tell you about the results of that. So what we used is Ken had made this wonderful stock with the ICRE transgene, and it hits a site in the repeated rDNA locuses. And so you heat shock it, and it's incredibly effective. You get almost 100% broken chromosomes. So it's a really good way of getting double-stranded breaks. And we can stain with H2AV, and we can see that they indeed do make double-stranded breaks. So the system we use to do this live is the third instar larva neuroblasts. So all we have to do is take the larva, we have them labeled with our favorite GFP markers, and these divisions are about an hour, so they're really nice to look at. So I think I have a movie here. Yeah, and you can see whatever you want to see in this. And so that's the system we work on. And so now, what we did then is first, okay. So first what we did is just looked at the neuroblasts and other cells as well. And here's what they look like. If the system works as advertised, here's a control anaphase. If you cut it with ICRE, you get the X chromosomes right in the left on the metaphase plate. And that's 20% of the genome. What was amazing is 80% of the anaphases looked like this. The flies were perfectly fine. So how could that be? Yeah, so that just says that. So you're losing 20% of your genome, the entire X, and you're fine. So how was that? And let me just show you this movie, because it really it's basically the answer to it. So just watch this. Watch these acentrics. So they do not have and I should point out, we stain them with all the centrosome proteins, both GFP and antibodies. They do not have a centromere. And in this talk, I'm going to switch centrosome and centromere many, many times. Just know I'm talking about the centromere. Yeah, Larry would not be pleased. So this was extraordinary. So this does not have a centromere, and yet they segregate late, but perfectly fine. So how is this? What is the mechanism that's doing that? Yeah. So brought up some questions. How do you drive these things poleward without a centromere to connect to the microtubules? The other thing is they're going really late. They're going so late that the nuclear envelope in telophase has already formed, so they're locked out. Right? So how do you deal with that? And then in the other thing, these I don't have the answer. I'm going to give you the answer to the first two. I don't have the third one, but how do you get accurate sister? These separate, the acentric sisters separate accurately. How do you do that? We do not know how you do that. I have some. We had some insights in the other one. So let me just show you this movie. What you'll see This goes on for a while. I have to find where there's the acentric. It's popping around there. And let this one go. And then there's this, and you get this little thread of DNA. And so we didn't really know. We go, wow, there's this little thread of DNA, and is that just extra DNA hanging over or lagging chromatin? What convinced us that this was more than just lagging chromatin was the fact we could stain with BubR1, which normally, in a normal, will just label the centromeres, and then a telophase goes away. What we could see is when we induced ICRE, we'd have this ectopic localization of BubR1 on these we call them tethers, these little threads of DNA. And that's shown here a little bit better. So here's a movie of, so let me get here. Okay. Here's a movie of just control, and that's what BubR1 normally looks like. Now here's a movie when we do ICRE, and we just stop it there. Yeah, so you can see that's where the tether is, and there's this ectopic BubR1 in here as well. So we went through, this was work, a really wonderful postdoc, Ann Roya, and she looked at other proteins. BubR1, Polo, Incept, and Aurora B are all on this. So this is it has DNA, it has histones, and these proteins on it. So we called it a tether, okay? And so what is the function of the tether? And the obvious thing to us was it was like a rope, and that it's just being pulled in, right? That's what you pull this thing into the daughter nucleus. And then I had a graduate student test this out, and just a really nice series of studies. If that's true, if this tether is acting when you don't have a centromere, it's acting as a backup mechanism, and you're just pulling this thing in, you should always have the telomeres lagging, right? And so we wanted to look at that. Is it the case that the telomere is always lagging, or is there a random orientation to this? Because if there's a random orientation with the telomere leading, it would be hard to believe the tether could be supplying the force. So that was the idea with that would be hard to believe would be involved in force production. So Travis Carr looked at this, and we could label the telomeres now. And if you look at this, this is a clear one where this is going and the telomeres leading. And this is a nice movie of this. I really like this movie here. So if you watch this guy. So yeah, here we go. And actually, it spends a lot of time to flip around to get in the right orientation to eventually get in the nucleus. So it's really trying to get in there. So, but what that meant is, all right, stop, okay. So actually, when we measured these, it was random. So the orientation of this acentric fragment is random. So that meant the function of the tether was not force production to move this in. So then what was yeah, all right, enough. Okay, so what was providing the force? And an obvious thing is when you do this, the acentrics are encapsulated by microtubules. So the obvious thing is that they're using microtubule motor proteins and being driven down into the nucleus by motor proteins rather than the canonical connection to the centromere. So the way we did this, we collaborated with Sophie Dumont at UCSF, and she has a beautiful laser ablation system. And we just wanted to see if it's microtubules, if we laser ablate the microtubules, we should affect the behavior of the acentric. And so we did that here. And I'll just show you the stills here. But what happens, as soon as you ablate this here, and you can see it's gone, this and this pop back together. In the movie, you'll see it's pretty clear. So as soon as you do that, it pops back together. So there was a really tight connection between the microtubules and the acentric. Okay. Let me see here. Yeah. So to find actually the genes, we just, we had a nice screen because I said with ICRE alone, these flies survive just fine, and we could take mutants that survive just fine and look for synthetic lethality because these would be genes just needed when you have an acentric and to get that acentric into the daughter nucleus. So we did that, and out of these we got 3 genes, and I'll just focus on this one. It's a chromokinesin. That sounds pretty good. It's associated with the chromosome. It's a motor protein. So we looked at that, and here's wild type. So now here's this KL3. So here's wild type again. If you look at that, there they go, and they're late, but they eventually go in and they get right into the nucleus. And then here's this chromokinesin background. You can just see it's not happy, and that's they all go into that. So it's aneuploid. So this one got all 4. This one got none. So that's aneuploid. So basically, we think, the solution to this then is it's using motor proteins rather than a centromere to get in. All right, so microtubules and motor proteins. So then that still leaves, we still have this tether. And what is the function of the tether? Okay, so so basically this leaves what's the function of the tether? And then how do these acentrics actually enter the telophase nucleus? Because they're way too late. The nuclear envelope has formed. And that's actually, those two questions go together. And you'll see this. So I have a great current graduate student, Brant Orwicki. And so he started looking at the lamins on this. And so so now the green is the lamin and the red is the microtubules. So now, here's wild type. And here you go, and you can see him, and you can just see you get these nice lamin formations. All right. So now we did this. We did this when we reduced I-Cree. Let me see if I can stop it. But you'll start to see there's a little a hole there, and that's that little channel where the lamins opened up, and these are getting in. And when we look at, we've gone to Genelia Farms with this and gotten higher resolution pictures, but when you start to look at it, you really see this is about this hole in the nuclear envelope about 6 times bigger than the nuclear pore, so it's not some just modified nuclear pore. And you can see this hole here, and here's the acentric entering in it. So this was really interesting. So basically, we're getting this localized delay in nuclear envelope assembly. And this comes back, I said on the tether there were 4 proteins, and one of them is Aurora B. And Aurora B is famous in biochemistry and in a lot of genetic experiments for inhibiting nuclear envelope assembly. So the idea was, you have this tether and what the tether is doing is making a localized concentration of Aurora B, and that's making a localized inhibition of the nuclear envelope. And so we can test that by disrupt. So here again, you have where you have the wild type, but with acentric. And now here's with let me stop it there. And so 2 things. So now this is when you've knocked out Aurora B, either RNA, and actually we have hypomorphic mutants that do this with a drug. So 2 things. It's locked out now. So not only the acentric doesn't get in, but you'll notice now the acentric is coated with the nuclear lamin. And in this one, it's not. So they get locked out. This forms a micronuclei, and then bad things basically happen. So yeah, and we can count these. And so what we think then, the model is that early on, there's other work doing about this global gradient of Aurora B, and this is how you time or position the formation of a nuclear envelope. You have Aurora B at the mid-zone here, and it's a gradient, and as soon as the chromosomes move off that mid-zone, they're away from this inhibiting factor of Aurora B, and you form a nuclear envelope. We think that model's true, and just what happens here is what we think is now on top of that model, we just have a localized, we add this little extra localized concentration. So even though you're far away from this main gradient of Aurora B, you have, and you make this, and the acentric can enter. Yes, and that's, so basically we have these, and I should say, we're now doing, we're looking in mammalian cells, and we had some pretty good hints that these tethers are present. These are X-rated mammalian cells. These tethers are present as well as there. So we think that the tether does two things. One is it maintains acentric so they can actually get captured by microtubules to start this process. And then more importantly, at telophase, the tether prevents nuclear envelope formation, inclusion into the daughter nucleus. So that's, and just I'll thank you, Larry. And he's a scholar, a teacher, and a scientist, and just a wonderful mentor that for all of us. Thank you.